Hi, this is Dr. G. Bailey, and I'll be talking on this video about the origin of cells, which is topic 1.5. Some of the things that you need to understand from the IB program. The first most important thing is that cells can only be formed by division of pre-existing cells. If you have watched the video on topic 1.1, you will see that I will talk again about Louis Pasteur and how he came out with how cells are coming from pre-existing cells through his chicken broth experiment. The second thing, you must understand the cells must have arisen from lone living material. This is where we're going to talk is how they became up with that first cell. Because again, Louis Pasteur says, yeah, cells of today came from pre-existing cells, but what about the first ever cell? We do have a theory, and we're going to talk about that theory through the experiment of Miller and Urey, how he came up with some of these ideas. We're going to understand the process of it. We're also going to talk about how it came to arise the origin of the eukaryotic cells. And we do this by talking about the endosymbiotic theory. So we're going to talk more about how eukaryotic cells basically evolved from prokaryotic cells. And then eventually, again, talking about Pasteur experiments and how he disproved the spontaneous generation, and talk about other organisms that do not occur on our Earth. We're just basically going to talk about his experiment again. So this is what the topic 1.5 is about. So let's start with the origin of cells. And when we say origin of cells, we're talking about the first ever cell. So we are basically saying that all the cells, they come from some pre-existing cells. But, okay, we can prove that through the Louis Pasteur experiment. But what about the first one? They have an idea that it was about 3.8 billion years ago. And they have some kind of assumptions, which we call this as a hypothesis. So the first group of people to come up with this hypothesis was the Operin Haldane. These guys had a hypothesis. And they had 4 statements in which they believe on how actually the first cell came up to be. The first thing they says is that there has to be some kind of formation of monomers. What do you mean by monomers? Small, simple units like an amino acid, a nitrogen gas, fatty acids, simple sugars. The formation of these guys, because these are the beginnings of everything. The second thing, it is the formation of the polymers, which that means we take the monomers right here and we create bigger things like polysaccharides, proteins, and RNA strands. Now, a lot of this information that I'm giving might be like completely clueless for you if you haven't touched topic 2 yet. But if you have touched on topic 2, then this will start to make more sense of these words that I'm saying. Then there's the formation of the nucleic acid polymer, such as RNA. And RNA carries hereditary formation. And the last thing is the formation of protocells. And these protocells, they carry all the metabolic reactions inside a closed system. In the case, it can be a cell. So basically, these are the 4 hypotheses that Operin Haldane says. What does that mean? They're basically saying that the first ever cell was only made of a plasma membrane like this. And inside of it, there was only a certain RNA, not DNA, just RNA. And that was pretty much it. A plasma membrane with RNA inside, and then from there, everything else was popping. This is what they believe that. So, looking this with more drawings, maybe you can get an idea. The first thing on the hypothesis they're talking about is how you have these inorganic molecules like water, CO2, ammonia, nitrogen gases, hydrogen gases, and methane. They were just floating in the atmosphere. Now, all of these are inorganic, which that means they contain no carbon, they're not living things, they're just floating in the Earth's atmosphere. But somehow, through the atmosphere of the Earth, 3.8 billion years ago, caused these molecules to eventually be converted. As you can see, all these molecules being converted or combining, turning into a bigger molecule. In this case, we're talking about monomers. So what I mean by monomers, we're talking about becoming amino acids, which we know that amino acids make protein, and nitrogen bases, which nitrogen bases can be part of making a DNA or an RNA, simple sugars, which can make a carb or can make DNA and RNA, and fatty acids, which can make so many things such as the phospholipid layer of a cell. So they went from inorganic material to become organic. Just by combining these molecules together, we form these other four. So it's all about synthesis, about making from simple inorganic molecules to becoming simple organic molecules. That was the first thing. The question is, how did you go from the inorganic to organic? They're basically saying something in the atmosphere caused them to combine. So by them combining these molecules, let's say all of these were amino acids, right? And what I mean by amino acid, I'm talking about all of these guys are these components. So a bunch of amino acids joined together through this process called polymerization, which that means adding monomers together. We got ourselves a large protein because protein are made of amino acids. And then this protein might have gone to replicate. That's one way. Or we can say the opposite. We can say that all of these particles right here is a nitrogen base and it's also made of some simple sugar, which caused this thing to be an RNA. So we just created RNA. And then RNA actually will go through self-replication. That means RNA will make a copy of itself. Now you have 1, 2 copies of an RNA. And then you could create molecules to make the membrane. So the membrane, if you remember, which is this is the membrane, what is it made of? It's made of lipid, right? And protein. And that's how you created that, by creating right here at this section. You created proteins, lipids, and you made the cell. And then the RNA is here, and voila, we have the first cell, which is just made of a phospholipid bilayer and RNA inside. That was the theory. So to make it more understanding, let's just say, if you still this not click, this would definitely click you. So you have, let's say, CO2, we have oxygen, we have methane, and these are particles, right? These are just little particles. I'm going to put them as little circles. And somehow they're hit on the Earth atmosphere. And the atmosphere turned these particles into some kind of structures. Now these structures, I'm just drawing for the sake of drawing. Don't think that these structures are perfectly right. These structures, this one could be an amino acid. And this could be a sugar base, a deoxyribose, we don't know. And then this formation. So this part here is inorganic. And these guys are actually organic material. Now these guys, they can join together and form things. So they can become bigger particles, coming joining like this, and voila, you could have made an RNA, or you could have made a protein, or you can make a lipid, whatever it is, a long chain. This part is called polymerization, which is just adding particles, small little particles, organic molecules together. Then maybe RNA start duplicating itself, and now you have a bunch of RNA coming out, copies. And remember, RNA can carry genetic material, so it can make other copies. And then somehow from this, we can also produce a cell membrane. And then from this, somehow we created the cell membrane, and inside we have, voila, RNA. And they were thinking that this is the first cell. So the process starts with inorganic molecules that turn into organic molecules. And these organic molecules combine within themselves and turn into large molecules such as RNA, protein, lipids. And they do this process through polymerization. And then the RNA joins with the cell membrane and turn into the first cell. Again, this is just a hypothesis. Is it true? We don't know. So then, because of this hypothesis done by a scientist, there was this group of two scientists known as Miller and Urey, which these guys, you do need to know their experiment with today. They decided to test the operandi hypothesis in their lab. They want to see if it's actually true. So they did. Here is the equipment that they created. And in that equipment, what did they do? They had different sections in which imitated what the story they were saying. And ironically, what did they find out? They were able to turn these inorganic compounds into organic molecules in their experiment. If you notice right here, in this drawing right here, you see there is electrodes which causes the lightning. So that means there was a lot of thunderstorms and lightning in the early times of the Earth. And they tried to create, the atmosphere vapor of water coming up. And they made all these organic molecules coming here and be exposed to an atmosphere of the Earth. And then they condense them, and then eventually they will figure out things that are popping out. Now, that's just a general idea. But here is the steps. So the first step is that they did was boil the water. So when you boil the water, it's evaporating as water vapor. Now, when it gets to water vapor, that water vapor is going to mix with methane, ammonia, and hydrogen gases. When they mix, then you put sparks of electrodes because you're trying to simulate the lightning of the atmosphere. Once you do that, then you cool it off. When you cool it off, they go back to being liquid water. And then from that liquid of water, you trap that liquid and you found out organic molecules were produced. So you don't have to do this entire drawing. You can just do steps. You can simply just draw if the teacher ever asks. First, you have a beaker and you can say boil water. And then this beaker somehow, whatever the water vapor that is here, right? The water vapor. And then here you inject what? You could inject methane. You can inject ammonia. You can inject hydrogen gases. And they will mix with the water vapor. So they're entering here and they're mixing with water vapor. And then they move to an area, right? In this area, you put electrodes. What these electrodes will do, they will spark, let me put here in yellow, they will spark lightning. So it's like the thunderstorms that happens in the early Earth atmosphere. And when these particles were exposed to this lightning, voila, you put a beaker here, right? And you're gonna condense them. Why you wanna condense? Because you wanna turn these particles who are like gases to become what? Liquid. That's what condense means. Condense means going from a gas to a liquid. And then these guys, once they condense, they become more liquid again, and then you come and pick up a bunch of liquids. And then what did the two, Miller and Urey, what did they do? They took these liquids here and they tested. And when they tested, they found out that they were organic. So they actually prove the first part. It just proves that, yes, you can turn inorganic molecules into organic. So this entire experiment was all about turning the inorganic to organic molecule. And that proves what? It proves this first part of the hypothesis. We're talking about formation of the monomers coming from inorganic molecules. So the Miller and Urey was able to produce that part of the experiment. So it concluded what? These organic molecules, which talking about amino acids, can come from inorganic compounds, which eventually these amino acids can produce proteins, which these proteins can produce RNA, and then boom, we have a whole entire cell being created. So looking back, we just mentioned about producing inorganic to organic molecules, producing RNA. What about the cell membrane? Well, if you remember the cell membrane, you know that the red parts here are basically the phospholipids heads, if you learned this from the previous topic on 1.3. And the blue parts right here, these blue parts are the fatty acids. So the blue part is the homophobic and the hydrophobic, and the hydrophilic here, it is basically the red parts. So having those two, they join all the possible fatty acids that were built up from inorganic to organic, and they become micelles. Micelles are like tiny little circles of phospholipid layers combined. So it's like small vesicles, and these vesicles made of phospholipid layers. So this is an example of micelles, and inside of it you have the RNA that was created from inorganic materials to organic, and then you have, of course, the plasma membrane, which is made of a lipid membrane. That's how they think it came out. Is it true? We don't know, but it kind of proves a little bit of that theory that there is a possibility that the atmosphere caused those inorganic material to become organic, and it just builds it up. But we don't know if it's 100% true. The other experiments that prove how spontaneous generation does not work and that cells come from pre-existing cells, it is Pasteur's experiment. Again, he's using a chicken broth, and he's through the chicken broth and trying to boil and sterilize the things, he was able to prove that living cells can only come from previous cells. It cannot come from something like nothing, from dead cells. So let me explain this process again in this video for those who don't understand. So the first step he did, again, is getting a flask, and then inside of it, we're gonna put its liquid. So let's say that this red thing here is the chicken broth. It's a soup made of chicken broth. And you keep it out there. What I figure out in the next days is that some bacteria were produced on the surface of that chicken broth. So that goes with the spontaneous generation idea, which it means things just pop out of nowhere, just pop out of the blue. So that proves the spontaneous generation idea. Things just pop out of nothing, out of the blue. So what he did, the second thing he did was he took this flask and he decided to put fire on it. So what he did, he sterilized. Oh, let me write this again. He sterilized. That means he boiled it and he cleared it. So when he cleared this up, now the soup or the broth is clear from all bacteria and fungus. There's nothing there. What's inside of it? Inside of it, there's only dead cells. There's nothing living. Everything is dead. That's after sterilizing. And he kept the flask open. After a day, look what happens. Here, the chicken broth had, again, those bacteria's growths happening at the surface. This proves again the spontaneous generation idea that, see, there was dead cells, and that means out of nothing, new things pop out. So this process, it proves the spontaneous generation is right. But then he came for the second idea. He's decided, okay, let me do something else. I am going to take this flask and I'm going to put the chicken broth and I am going to sterilize it with fire. But here is the interesting thing now. I am actually going to close the flask. I'm going to close it so nothing can go in or out. I am going to sterilize it and see what happens. He did this. And after a few days, he go back and he looked at the soup, which in this case now it was closed, right? It's been sterilized. If it's sterilized, that means there's only dead cells inside. So inside, there's only dead cells. And after first day, second day, third day, remember, it is closed. Nothing is showing up. No bacteria are showing up. Nothing. So dead cells continue to be what? Dead cells you didn't see any new cells popping out. You didn't see anything living popping out. This part with the closing flask disproved the spontaneous generation, because what did Pasteur say here? Hey, I just proved that things cannot just pop out of nowhere. Because if it is true, when I close this flask right here, after a few days, there should have been bacteria popping out, but no bacteria popping out. And since there's nothing that can go in, like it's blocked, right? Nothing can go in. Dead cells continue to be dead cells. I didn't see any living cells just popping out of the blue, popping spontaneously. So when he closed it, he disproved the spontaneous generation. And they're like, okay, if that is true, then how the hell do these bacteria just show up out of nowhere? So he's like, you know what? Let me show you how. If I break the flask, now I let the air comes in. And like he was saying, in the air, you have bacteria floating in the air. So now that you open the flask, you allow the living cells to come in and land on the surface of the broth. And since living cells are landing on the surface of the broth, now new cells are being produced, and now you have a bunch of bacteria growing, producing on top of the surface. Which this proves what? That cells can only be coming from previous cells. Which previous cells? These were the previous cells that landed, and then they start to continue. So the living cells can only have come from pre-existing cells. Again, from living cells. So nothing just pops and arises out of nothing. There must have been a cell before, and that cell divided and caused a new cell to show up. And then that cell divided and caused the next cell to show up. But it was not out of nothing, thin air. So Louis Pasteur experiment disprove, rejected, spontaneous generation. He says, dude, there's no freaking way that things can just show up out of nothing. There has to have been a living cell living there before, and that cell divided itself and created more living cells. So cells do come from previous cells, and they do not come out of nothing. So this is a perfect experiment to prove that. Now, there is another theory which is saying, how did we go from prokaryotics to form eukaryotic cells? Now, this is a woman, and they made this theory. She explained that if you see mitochondria and chloroplast, interestingly, they have the same size as a bacteria. So if I draw mitochondria right here, it has made of two layers, right? This is a mitochondria. And if I decided to draw a chloroplast, the chloroplast would also be two layers, right? And it has its stacks here. It's still the same size of the mitochondria. And then here comes the interesting part. A bacteria still has the same shape, and it also is made of a cell wall that is made of two layers. And the three of them, the bacteria, the mitochondria, the chloroplast, and the bacteria, the three of them have exactly the same size. Interesting. The next thing is that the three of them have DNA. See, the mitochondria has its own DNA, the chloroplast has its own DNA, and the bacteria has its own DNA. Another interesting thing also that the three of them have is that mitochondria has ribosomes. And guess what? Chloroplast also has ribosomes. And guess what? The bacteria also has ribosomes. That's another thing. And interesting, when you're going to make a new, for example, if you want to make a new mitochondria, it does through binary fission. That means it produces another mitochondria by making a copy of itself. Same thing here, it does by binary fission. So you can make another chloroplast by just copying its own self. And guess what the bacteria does? It also does binary fission. So far, the three of them have DNA. The three of them has double layers. The three of them divide by binary fission. The three of them have ribosomes, and the three of them have the same size. How interesting is that? That means that somehow, back in the old days, a mitochondria was actually a bacteria. And it's most possibly that the chloroplast was also a bacteria. But, so where does this all come to? Basically, let's say there was some kind of harsh environment around, right? You actually have a prokaryotic that created himself a nucleus, and then because of the harsh environment, there was like a mutualistic relationship, which means we both benefit. You help me, I help you. So what happened is, this is a chloroplast, which back then could have been known as cyanobacterium, and this was an aerobic bacterium, which is the mitochondria. They both joined with this prokaryotic here through this thing called endosymbiosis, which means let's come together, let's join our forces, let's help each other. And then voila, you created an eukaryotic. So what happened is a prokaryotic cell engulfed, or should I say ate, in a sense, like allowed the chloroplast and the mitochondria to come in, and together they were able to survive the harsh environments of the atmosphere of the earth, and which led to the new eukaryotic cell. So as a wrap up, where do we come with this evidence? Like, is it really true that a prokaryotic cell went there and decided to take a mitochondria in and decided to take a chloroplast in? Well, it's possible. They will say, how is that possible? Well, there's 5 evidence that show that there is a huge possibility that a prokaryotic eventually digest. I shouldn't say digest because it means it's eating. I would say the word engulf. That's the right word, engulf, because engulf means you don't eat it, you just bring it in. So engulfed both mitochondria and chloroplast. What is the proof? They're membranes. I told you, they're made of double membrane. Another thing that I have not mentioned is antibiotics. Do you know that if you put an antibiotic pill, you are destroying bacteria? But guess what? You can also destroy a mitochondria or chloroplast with antibiotics. Again, mitochondria and chloroplast acting like a bacteria. They cannot resist antibiotics. They die with it. All of them, they're doing by binary fission. They all have division through binary. They all have their own DNA. And they all have a ribosome that is a 70S. And if you remember, 70S is the size for what? For prokaryotics, which again, most likely the mitochondria and chloroplast was actually a bacteria. So these 5 evidence here might just prove that mitochondria and chloroplast, they used to be bacteria and they were engulfed by a prokaryotic, and then when they were engulfed, they turned into an eukaryotic. So this is pretty much for this topic on 1.5. Here you have all the questions. You can read these questions, go back to the video and find out the answers. And this is all for this. Thank you.